You're all set.
Hello, my name is Grace Kistner. I'm a senior at Bridgeport Regional Aquaculture Science and Technology Education Center, and I will be talking about utilizing the nematocysts of Cassiopeia zimicana as a bioactive anti-proliferative compound. Can you see the slide changing?
I
No.
cannot see the slide changing.
Okay. So what is the issue? Many anti-proliferative and antimicrobial products contain corrosive chemicals such as ammonia and hypochlorite that have been linked to various respiratory and dermatological issues. Why should you care about this problem, and why do I care about the problem? So I intend on using Cassiopeia zimicana as a safe alternative to these harsh cleaning chemicals, because most of these chemicals can cause harm to human and animal health as well as the environment. And as you can see over here, I have this graph that just shows the different issues that can cause a lot of dermatological issues such as itching, redness, scaling, respiratory issues such as shortness of breath, burning in your chest, and it can actually cause a lot of adult onset asthma, as well as when you're using these products such as like wipes or sprays, the particles can get released into the atmosphere and cause volatile organic compounds, as well as ground level smog, which can cause disease in plants, as well as inhibit seed production, which basically means that seeds are not able to grow to their full potential. And these chemicals can also be exposed to aquatic life in untreated water, such as sewage water. So what am I going to do to solve this issue? If cultures of microorganisms are inoculated with Cassiopeia zimicana nematocyst toxin, it will then serve as an anti-proliferative/antimicrobial agent, resulting in reduced or no cell growth. Now, what are Cassiopeia zimicana? This is a species of jellyfish more commonly referred to as upside-down jellies or mangrove jellies. They're called upside-down jellies because instead of floating through the water column like regular jellies, they rest on their bells on the bottom of mangrove forest beds. Now they are able to get their food and nutrients by creating a self-regulated water flow by pulsating their bell and bringing water towards them. 90% of their energy is actually provided by a symbiotic algae called Symbiodinium microadriaticum that grows in their tentacles. So the relationship is basically that the algae provides them energy and they provide the algae a safe place to live. So in the previous slide, I mentioned their oral arms, which is what they use to eat. This is where they also store all of their toxins. Their nematocysts reside in their oral arms, and these are basically the stinging cells that all jellies have that host their toxins that they use to stun their prey. So instead of a normal jelly using their tentacles to sting their prey, these jellies will actually release structures of mucus called casiosomes, which have an outer epithelial layer of these nematocysts. So when the mucus is released and it comes into contact with their prey, their nematocysts are fired off and their toxins are released. Now, each class of jellyfish has different types of toxins. These jellies in particular are class Scyphozoa, meaning that their toxins consist of phospholipase B2 and metalloprotease, which are both enzymes, and they also have pore-forming toxins, which is a type of toxin that spans across every class of jelly. So as I mentioned, their nematocyst and casiosome, I have a diagram right here of the development of a nematocyst. So basically when a nematocyst is fired off, it has this coil inside of its structure that when it's fired is sprung out, and it has barbs that will release the toxins into whatever prey it is trying to consume. Now, next to this, you can see is a casiosome, which I mentioned is where the nematocysts reside. This outer layer is basically filled with their nematocysts. I also mentioned the toxin phospholipase A2. I really tried to hone in on this toxin when I was doing my research just because I found its properties really amazing. This toxin uses convergent recruitment to assault its prey. Now, this basically means that when a nematocyst is fired by the jelly, it carries over proteins with it that help it execute a more impactful attack. And this toxin also helps to aid in the innate immune system in killing bacteria. Now, I really found this interesting and a perfect property with what the research I was doing was because I wanted to use these toxins to stop the growth of or kill bacteria, so it lined up perfectly with my research. So, how did I get the mucus from the jellies? I started off by taking the jelly out of the tank and placing it in a beaker with its tank water. I then took a disposable pipette. I took some of the tank water in the pipette and I gently pipetted it onto its oral arms, which then stimulated its production of mucus. I then used another pipette to collect the mucus out of the water and store it in a separate vial so that I could separate it from the tank water. And I then centrifuged that mucus sample for 30 seconds at 5,000 RPM. This was so that I could separate the tank water or the supernatant away from the mucus to get a more concentrated pellet sample. And as you can see here, this is what I'm doing in that photo. So to evaluate the antimicrobial properties of the mucus, I used E. coli as my bacterial model organism. I started off by launch streaking E. coli onto 6 different plates of nutrient agar, and I incubated all 6 plates at 37 degrees Celsius for 48 hours. I then pipetted 25 microliters of the mucus sample only onto 3 of the plates, and then I used a sterile swab to swab it in a 1 centimeter diameter area, just so that I could have a uniform section across all of my plates that I could evaluate. I then took the remaining 3 plates and I pipetted 25 microliters of just the plain tank water onto them to use as a control, because one of my concerns was the salinity of the water affecting the E. coli, and I wanted to be able to rule that out as a factor and determine if it was only the toxins and the mucus affecting the growth. And then I put all 6 plates back in the incubator for 24 hours again. And when I took them out, I observed it for growth in the swabbed area. And as you can see here, this is my control plate. I swabbed it right here in the middle. And as you can see, the E. coli continued to grow after being incubated again. So I was able to rule out the salinity of the water affecting the growth of the E. coli. So for my results, as you can see here, there's absolutely 0% growth in the swabbed area. I mean, there's not much to say about it because it's kind of like a yes or no. This photo is just a zoomed in of that section right there, so you can see it better, and I circled the area. And as you can see, it has clearly defined contact margins between the colonies and the treated area, so the E. coli did not intrude in on the swabbed area at all during the 24 hours after it was incubated. So I was able to determine that the mucus has very efficient antimicrobial properties. So to evaluate the antiproliferative properties of the E. coli or of the mucus, I used yeast as a model organism for eukaryotes. So I started off by mixing 5 milliliters of YPD broth and 15 milliliters of distilled water with 0.1 grams of yeast in a beaker and mixed it all together until it was completely dissolved. Now, for my test of the mucus, I added 500 microliters of the mucus to that sample. And then for a control, I again added 500 microliters of just the tank water to rule out the salinity affecting the growth. So I placed both of these in a 33 degrees Celsius water bath, and I did this to basically stabilize the temperature because yeast is very picky with how it grows in different temperatures, and I didn't want my results to be all over the place because it was too cold or too hot. I then created a blank for my spec 20. I made that using 3 milliliters of distilled water and 1 milliliter of YPD broth. And as you can see here, this is what the blank looked like. I placed my samples in the spec 20 for every 10 minutes for 1 hour at the absorption of 600 nanometers. And I did it every 10 minutes over an hour just to see the progress of its growth. And for my sample, I made this using 1 milliliter of my yeast solution that I described before. I mixed that with 3 milliliters of distilled water in a cuvette. I then took out 2 milliliters of this above solution and replaced it with distilled water to dilute it because the spec was not able to properly read it with such a high concentration. So I just basically made it easier for the spec to read. So for the results, I calculated a 6,259,720.25 cell count difference from the control to the treated, meaning that that was that much less cells in the treated compared to the control. This then translated to a 66.2 decrease in the growth. And as you can see here, I plotted all of my points. This top green line, that is the control. As you can see, it has sort of exponential logarithmic growth. It hit its peak at 50 minutes, and this one down here, that's just the log depth of the sample because it doesn't last for very long. And then as you can see down here, here's all of my trials. You can see a consistent static population in the treated sample compared to the control sample. And then to test the strength of my relationships in my data, I calculated the R values for all of my trials as well as the control. So as you can see, the control has an R value of 0.7, meaning that it has a very strong positive relationship, which is demonstrated by how it looks in the graph. And then all of my trials either had an R value close to 0 or closer to -1, which is great. It's just what I wanted. It means that basically the treatment of the mucus caused like the population to be very static and not grow, or it either made it decline in growth, which is
2 minutes.
like how I expected it to go. Now, for my conclusion, so my results 100% proved my hypothesis. Everything went exactly as I had expected it to go, which is wonderful. It clearly demonstrated anti-proliferative and antimicrobial properties in both the E. coli and the yeast. There was, again, 0% growth on the soft area of E. coli and a 66.2% decrease in the yeast cell density, which is amazing. It went perfectly how I wanted it to. And then also I wanted to just address a concern that I got in the comments of my video that I submitted. And it was a concern about frequent use of this product, even though that this product poses no health risk. It is recommended that gloves, goggles, or any other PPE are used as with any other grain cleaning product. And use of this product is very comparable to washing your hands or using a hand sanitizer, in the sense that it will still affect your normal human probiotic flora on your hands. So I just wanted to address that because I did get a comment about it. And then these are all of my references.
Okay,